Hello, everyone. My name is Aricia Stus, and I'm actually an undergrad bioengineering student here at UCSD. And today I'll be speaking on behalf of Engineering World Health on low-cost paper-based genetic circuits for detecting HIV. And I should use this. So Engineering World Health, or EWH, here at UCSD is a completely student-run organization. We're also a nonprofit that focuses on allowing students to use different kinds of applications of engineering and apply them outside of the classroom. And our main project goal is to design and build low-cost technologies, whether they're mechanical or biological, to detect HIV antiretroviral treatment or ART failure in patients of developing countries. So HIV, or human immunodeficiency virus, as you all know, is a retrovirus which pretty much hijacks the immune system of inflicted patients by binding to the T cells and depleting the T cell population in the patients. If untreated, it results in AIDS. Currently, it affects approximately 37 million people worldwide, and according to WHO, it's the 6th leading cause of death in the world. Now, if we zoom in on just low-income countries in general, it turns out, according to WHO, to be the 2nd leading cause of death in the low-income countries. How HIV works is, again, it's a retrovirus, and it pretty much binds to CD4 target sites on host cells, allowing it to have a membrane fusion with the host cell and uncoding and releasing 2 copies of its viral RNA. This viral RNA is then able to actually integrate into the host genome and hijacking the genome or hijacking the host and allowing the virus to replicate, eventually killing the cell and spreading throughout the patient's body. Now, time of detection is very much key when dealing with HIV. When patients are first diagnosed with HIV, they're immediately placed onto a combination of drugs. The reason being, it's been shown by numerous studies that if no kind of therapy is implemented, that the patients within a couple of months really progress towards AIDS and eventually death. And currently, a lot of the diagnosis is in use of CD4 levels. Now, although that is a very reliable test, we also want to note that viral load testing is also a very feasible and also a much better detection system in many regards, because it allows you to actually detect the progression of HIV at a sooner position in time. Now, HIV viral load, what it pretty much is you're quantifying the RNA copy numbers per ml in a patient's blood. And ideally, you want an undetectable amount of this HIV or RNA copy numbers in your blood, or else the virus is actually progressing. But this viral load test requires access to laboratories and facilities, equipment such as centrifuges, thermocyclers, antibodies, which also require refrigeration, and the hands of a trained technician. And now when we're looking at different types of facilities in the developing world, not all facilities have these kind of technologies and equipment available to them. And that's why we want to actually create some kind of a device or technology that takes that into consideration and allows some kind of a test that would help all these clinics and patients abroad. Antiretroviral treatment, or ART, is pretty much a cocktail of drugs that inhibit the replication rate and integration rate of HIV. It's intended to keep HIV RNA copy number at an undetectable level. And there's 2 lines currently. The first line and second line both of which are used. The majority of patients use the first line. Say about 6% of infected HIV patients are turned to the second line due to no more response to the first line of ART treatment. And the reason being is because HIV-1 is a very highly mutagenic pathogen, being one of the most, having one of the highest mutagen rates or mutation rates for any pathogen. So it's very important to constantly monitor the HIV RNA viral load in patients, with tests being typically done every 6 months. And as you can see by this diagram, here's a suggestion about if a certain type of copy number is detected, to continue with the type of treatment that you're currently on, or eventually initiate second-line ART treatment in order to try to get back to the lower undetectable numbers of HIV RNA. Now, at our organization, we asked, how can we create a low-cost system that can rapidly and reliably test for the presence of HIV viral load? And we hope the answer is actually in synthetic biology. Now, the reason we make this statement is because of this paper by Keith Barty on his work on paper-based synthetic gene networks, where he was able to take, take gene circuits and actually apply them in detecting RNA from Ebola. He was focusing on different strains of Ebola because, as well, Ebola does contain high mutation rates as well. And he took trigger RNAs that were highly conserved from the Ebola virus and created a switch RNA containing a toehold region that, when complemented with this trigger RNA, would linearize, exposing the ribosome binding site, and thus initiating a downstream effect of transcription and translation, and eventually outputting either a fluorescent or a LAC-Z-mediated color change. And now he went a step further and was able to show that using these synthetic gene networks, he was able to, in combination with DNTPs, tRNAs, amino acids, buffers, as well as the necessary enzymes for transcription and translation, put this combination mixture into a 2 microliter reaction and onto a small piece of paper, freeze-dry the piece of paper, and he was able to show that this freeze-dried piece of paper remained stable for up to a year at room temperature, and upon rehydration with a trigger RNA, was able to output either a GFP reporter signal or a LAC-Z-mediated color change on the piece of paper. Now, this got us very excited because not only is this cool science, but it has so many implications in actually detecting HIV viral load. One, it allows us to use lower volumes of reagents to actually test for viral load, and thus lowering the cost per test. 2, freeze drying eliminates the necessity for reagent refrigeration. And 3, colorimetric output can be seen with the naked eye, can also be quantified and give you a reliable, or hopefully a reliable reading for viral load. So taking this paper as inspiration, we wanted to apply what Keith Barty had done for Ebola, or how he used Ebola for his system, and we wanted to do the same for HIV. And our first steps were really just to determine which target regions we wanted to consider in our trigger RNAs. What we had done was, considering the RNA genome of HIV is vast, we chose to focus on the GAG polygenes. The reason being, these 2 genes are very necessary for the replication of the virus. We took different kinds of strains of HIV and did alignments for these strains, and were able to determine about 21 to 24 base pairs of highly conserved regions from our alignments, and furthermore, we were able to also confirm our findings with published work for the highest conserved regions of the GAG polygenes. And based on our findings, we began to design our trigger RNA sequences, just as Keith Barty had done. Our switch designs relied on a switch or on a toehold region that really needed to complement or would have a high complementation probability with our trigger RNA. And we also created a positive switch region, or just positive control, which didn't have a toehold region and didn't depend on the presence of a trigger RNA to spark any kind of a laxi-mediated color change on the piece of paper. We had constructed a multitude of switches, but in order to actually gear down and focus in on which ones we wanted to test, because again, we're students, we don't have that much funding, we chose to run different kinds of probability tests using open source software NUPOC, which focuses on providing free or minimal free energy diagrams of the secondary structures of any type of RNA or DNA sequences that you implement. It could be one or more sequences at any kind of concentration that you're inputting. And from this, we were able to first test how our switches had looked, and based on the highest probability of complementation, and we were ideally looking for structures like this where we had a very nicely preserved toehold. Next, we wanted to test, well, in the system, how is the switch and trigger going to work and complement to one another? And we were, again, looking for high rates of complementation between the toehold, or between the toehold switch and the trigger above here. And from our testings, we were able to isolate and localize on 4 different kinds of regions that we wanted to focus on for actually constructing and assembling our circuits and further testing. So once our switches were assembled, and our triggers were amplified, we did a gel electrophoresis confirmation for the size of all the sequences, as well as sequencing validation. Specifically, this one is for switch GAG 545. So far, what we've been able to show is that our positive toehold does work within an hour of incubation at 37 degrees on a piece of paper, similar to what Keith Barty had done. We're currently in the process of optimizing our system for the trigger and switch reaction in complement to one another. Ideally, what we want is to optimize a system where we can obtain a reaction within 1 hour, quantify reliably, and scale up to the entire RNA extractions from whole blood samples. Now, the reasons to scale up, and actually a difficulty that will come, and in Keith Barty's paper, he was able to show that he could get complementation using trigger RNAs. But he hasn't actually, and no one to my knowledge, has been able to take actual genome samples and implement these gene circuits, at least for HIV. And here we have an MFE diagram showing our entire toehold switch and the trigger RNA. And again, you have pretty good high complementation based on the equilibrium probability. And how we actually hope to integrate the genetic circuit into EWH's overall system is we currently have a couple devices that have all been student-made. The first one being a centrifuge. What we hope to do is take a blood sample from a patient, input it into our centrifuge, and separate the blood into 3 layers. Next, we hope to isolate the plasma sample from the blood, input into our RNA extraction microfluidics device, which requires minimal technician skill and is very much automated, and gives you a very good eluted, concentrated amount of RNA. Next, from our eluted RNA, we hope to rehydrate our genetic circuit grids and based on some kind of a standard, create some kind of an algorithm that would be able to quantify the viral load based on pixel intensity as well as temporal dynamics within the 1 hour of our reaction. Finally, implementation of our technology is important. we, as engineers, we want to see our products actually work in the hands of some kind of clinics. And we're fortunate that we are paired up with a clinic called Health Frontiers Clinic in Tijuana. And they've expressed a very high need for this kind of, or for ART monitoring, because currently they don't have any kind of system that would be able to do so. If you look at the price breakdown of our system compared to that of what's currently available or maybe used in clinics here in the US, our system is by far a lot cheaper in comparison, and we do truly hope and believe that in the use of, or by using synthetic biology, we can create a reliable test. Finally, I'd like to acknowledge some of the students that I've been working with. Jahir, Chris, and Neil, they're all, well, two of them are undergrads. Jahir was our mentor for this project, still is. And also our sources of funds. Thank you, and I'll gladly take any questions.